So I think we have a few different ideas about what a mutation is. A mutation, strictly by the definition, is a change in the DNA sequence. So the root word for mutate is really just to change. And we often think about a change in the phenotype, but technically the mutation is a change in the DNA sequence. And we'll see that sometimes that contributes to phenotype, and sometimes you can have a DNA mutation that does not cause any difference in how the organism looks or functions. So the second question here, the effect of protein, effect on the protein, if there's a mutation in the DNA, the short answer is that it depends. It really depends on what that DNA change did relative to the process that is required to make the protein and the function of that protein. So we'll see lots of examples of mutations all the way from having hardly any effect or no measurable effect, all the way up to having severe effects on the organism that has those mutations. So an example here from your textbook of the CFTR protein, this is a membrane transporter, and the CF part of that is that this protein is implicated in the disease known as cystic fibrosis. So cystic fibrosis can be caused by this single protein having a mutation that prevents it from doing its normal function. So on the left here is the normal functioning CFTR transport molecule when it's pumping ions outside of the cell. And the mutated version of CFTR is unable to do that. So it's got to change to some part of its amino acid sequence that prevents it from doing that pumping job, and there's a buildup of mucus outside of the cell, and that generates, that all contributes to the phenotype. It's a major life-affecting phenotype of cystic fibrosis. So it comes down to one protein and potentially just one mutation to make that change. So this would be an example of a rather extreme effect of a mutation. Some other examples, so we don't get into protein function in this class, but I think we can probably safely try to understand what proteins do. So they have jobs interacting with other molecules. That's one of their big jobs. And the interaction with molecules, as we've seen, requires sort of protein biochemical signatures at different parts of these proteins. So some examples here on the left is a set of phosphorylation sites. Phosphorylation is a process of adding phosphate groups to proteins, and can be used, you can think of it sort of as activating proteins. So if you attach a phosphate group to a protein, that can be a signal for it to do something, can signal to other proteins or whatever. And so these shown on this diagram are specific phosphorylation sites and the molecules, the amino acids involved. So threonine 63, threonine 108, serine 353. Those are, in this molecule, very important amino acids relative to the rest of the amino acids. So if those got mutated, that would be a big effect, preventing that molecule from being phosphorylated at those sites. Other examples here, this is a molecule showing some active sites. These are the amino acids in their relative positions. So tyrosine 466, valine 484, and so on. Those molecules, those amino acid pieces, these are parts of the overall protein, those are interacting with this molecule called DON. And they need to be in their precise position and have their chemical properties to be able to hold that substrate molecule in place, interact with it, and so on. So if a mutation occurs at the active site, so if you mutate away one of those amino acids in the active site into a different amino acid, that might prevent this whole protein from interacting with its substrate, or mutating another part of the protein that lets it fold into the shape where it can interact with that substrate. And then other examples, we saw before cases of hydrophobic portions of proteins where they interact with membranes. That's a pretty common use for hydrophobic amino acids. So if those amino acids go from being hydrophobic to hydrophilic, that can prevent this molecule from staying or being located inside of the phospholipid bilayer. So sometimes rather severe effects, as we see. So the process of getting a DNA mutation, there are many different causes for DNA mutations. And one of those we explore in lab. We're going to do a ultraviolet light mutation. UV light causes um, nucleotides that are next to each other to what we call cross-link, and they during the repair of that error, they end up getting differences in the DNA sequence. There are other molecules. There are molecules known as carcinogens that cause cancer, or mutagens that cause mutation. They basically interfere with the cell's ability to copy its DNA correctly, and those can generate nucleotide changes. On the two examples on the right here show cases where um, not quite the correct nucleotide, not quite the correct nucleotide can be incorporated into DNA because the match is sort of good. So these are the bonds that are normally formed between cytosine and guanine. And if instead of cytosine, the guanine finds itself across from thymine, it can make hydrogen bonds. They're not optimal, but they're sort of almost acceptable. Same thing, normally T to A, sometimes C to A. So these are what I want to show you here is just that there are more likely differences to exist, more likely causes for mutation in these types of nucleotides. Because they're a little harder to detect. So the machinery, the polymerase, and other enzymes, they should be able to detect when there's a mismatch of the wrong nucleotides pairing with each other, but these are ones that sometimes slip through the cracks. So we'll spend a lot of time talking about the first kind of mutation here, single nucleotide substitution. So that's one nucleotide swapped out for another. That's the basis for those SNPs we've talked about. Insertions and deletions, and then in a later topic, we'll talk about chromosomal mutation. So we won't really get into that right now, but that's kind of a large scale mutation possibility. So probably if you've covered mutations before, you might have spent some time talking about the effects that a mutation has on protein coding DNA. So we'll spend a lot of time in this course emphasizing that not all of the DNA is coding for proteins. So not all pieces of DNA are made up of codons, but we will talk about them here. These are some examples of changes to the DNA that will get passed on to the RNA after transcription, and then that change of the sequence will be used as the codon for making an amino acid. So starting off at the left here, the original DNA sequence has a TCA, AGT, and transcription of that turns the TCA at the top strand into UCA, and that codes for serine. So a missense mutation, this category is results in changing a single amino acid. So by changing the middle nucleotide there from a C to a T, TTA becomes UUA codon, and that encodes leucine. So that single nucleotide change actually caused the amino acid that was encoded to become a different amino acid. And in this case, it didn't affect anybody to the left or right. These circles are the same. Here's a case where something could be more drastically changed. TCA in the normal case now turning into TAA becomes UAA, and UAA doesn't encode an amino acid, it's actually a stop codon. So whatever the protein is as it's being made, at this point it would stop being made. A very extreme change to the resulting amino acid sequence. And that's called a nonsense mutation. So missense turning one amino acid into a different amino acid, nonsense mutation turning a amino acid coding codon into a stop codon. And then this third one called a silent mutation would be a nucleotide change. And this is the case where it still counts as a mutation, but the TCG becomes UCG, encodes serine. As a result of that single change, it still encodes the same amino acid. So for the most part, that would have no effect whatsoever on the organism as a result. So good name there, silent mutation. So all of these A, B, and C, these are all single nucleotide changes and examples of how extreme of an effect they can have. So when we look at the effects here, you can kind of consider each category as to whether it has a major effect, a minor effect, or potentially no effect on the protein, the polypeptide sequence. And maybe we can expand on that and draw some examples of these. Okay, so first off, the missense mutation would end up producing an amino acid that's the same length. So generally, maybe a minor effect on the polypeptide. What might cause a bigger effect would be if that amino acid that gets changed, if that was one of the more important amino acids for holding the 3D structure together of the protein, for being part of the active site, for being a phosphorylation site. And those could be different kinds of changes that result from missense. Nonsense mutation, we typically think of having maybe a more severe effect. Let's go here. All right, so I'm going to draw just a series of circles, kind of like what your book has there for the amino acid sequence of the protein. All right, so if you have a normal case and you go in for a nonsense mutation, it's possible for that nonsense mutation not to have a big effect. Let's say we go here. So if the mutation is in one of the amino acids toward the end of the protein, then the protein that's encoded has all the same amino acids up to the point of the new stop codon. And if that stop codon was located at an amino acid that wasn't so important, and the amino acids after that were not so important, and there were not many of them typically, then that might be a change that was not a major change to the function of the protein. In contrast, if you had a nonsense mutation early on, that would cause here everything after the third amino acid just not to be made. That would be basically a missing protein, and it would almost certainly have no function, and that would be a major effect on the function of that protein. So generally in the nonsense mutation, the effect has to do with how early in the sequence that nonsense mutation cropped up. So how much of the wild type, the normal protein, was not made as a result of the mutation. Now, as far as the, let's see, we'll get into other kinds of mutations a little bit later. Silent mutation, typically no effect whatsoever. So we can think about looking at our codon table, the kinds of codons with the amino acids that they encode, and what kinds of changes could cause them to be different. So I'm going to get my annotator up here. Yep. So looking at the table, one of the patterns that we observe is that a single nucleotide change at the third codon position tends to result in a silent mutation. Why is that? Well, many of these are blocks of 4 codons that all encode the same amino acid. So if you took any of these codons for leucine, CUU, CUC, CUA, CUG, if you swapped out that third nucleotide for some other nucleotide, it would still be a leucine. So here, 4 that all contain codons for proline, 4 for serine. So looks like about half of these blocks, if you change the third codon, it would have no effect whatsoever, right? So that's owing a little bit to that wobble pairing that we talked about in translation, and also the fact that we've got these blocks of codons encoding the same amino acid. All right, here's a thought. Which ones of these could mutate to become a stop codon? So if you want to think about changes, the way this table is laid out, you could go left and right and up and down, and those reflect changes. So if you wanted to backtrack and say, all right, we got a UGA here, what kind of codons could mutate to become UGA? Well, everything in this block here is one letter off at the third position. So UGU could become UGA, UGG could become UGA, and that would generate this stop codon. Changing the second nucleotide position of the codon would change you in this dimension. So UUA leucine, UCA serine, those amino acid codons could become UGA by changing that middle nucleotide letter. And then moving down, UGA, CGA, AGA, GGA, these are 4 codons coding arginine and glycine that could mutate to become that. So for every one of these stop codons, you can kind of look across, up and down, and see where the changes could happen to produce those new stop codon. Here's another way to look at that. If you look at the AAG codon for lysine, what could it mutate to be? If you mutate the third position, one of those is possible to be a silent mutation, AAG to AAA. Those both encode lysine. Some of the changes at the third position would encode a different amino acid, asparagine. AAG, take it across the rows. So a change at the second nucleotide would turn that lysine codon into arginine, threonine, or methionine. And then moving up, AAG could become GAG for glutamate, CAG for glutamine, or UAG for a stop codon. So this is kind of a non-random pattern to what codons could become what other codons. Certainly you don't have to memorize any of that, but I think that's maybe a helpful way to explore the space of where mutations could happen. Now, as far as missense mutation, I mentioned differences that affect the active sites, phosphorylation sites, and so on. The chemical properties of amino acid, I think probably the easier one to think about is hydrophobic versus hydrophilic. And there's a term that refers to changes that have less effect or more effect on how the chemical properties of the amino acid change. So a conservative substitution is a term referring to a change that basically swaps out the chemical properties of 2 chemically almost equivalent amino acids. So lysine to arginine, that's a single nucleotide change, AAA to AGA. And on our graph down here, lysine, arginine, they have a lot of similar chemical structure there, at least relative to some. So they're both in this positively charged polar amino acid group. And also here we have basic amino acids. Non-conservative substitution. Here's another single nucleotide change, CAU histidine to UAU tyrosine. Here's histidine, here's tyrosine, and this involves going from a hydrophilic amino acid to a hydrophobic amino acid. That would have a pretty big effect on that amino acid. It might be in a hydrophilic region, and could destabilize that whole region for this change. So here's just an illustration from a paper that was pointing out the amino acid sequence of this protein, and as well as some known mutations that are differences in the amino acid sequence. And here's one that's possible. So alanine to valine was conservative sequence, conservative substitution there. Okay, so we've seen single nucleotide changes. It's also possible for pieces of DNA to be inserted or deleted. Sometimes this happens also as a mistake of copying DNA or for other reasons. But the end result is that the sequence of nucleotides shifts over some number of letters. So by the shift, this shifts the reading frame. And this becomes what we call a frame shift mutation. So the effect on the coding sequence is a frame shift. And the cause for that is an insertion or deletion of nucleotides. So I have that in insertion or deletion results in a frame shift unless, and the unless there is unless the insertion or deletion involves a multiple of 3 nucleotides. So yeah, if you added 3 nucleotides, you'd be adding a single codon and the remaining letters would stay in frame. Or if you deleted 3, you might delete a single codon and the rest of the letters would remain in frame. Let's see what happens with the result of the translation of this. So the original codon was GTA, would become GUA. That one is valine here. Change that to a G. Let's see, base substitution. We'll skip that one. Base insertion gets us GTT. GTA becomes GTT. GUA, GUA, GUU. Didn't change anything here, but it had actually a big change on the next codon. So the insertion of that T changed the next codon from a GAT to an AGA. And that effect would be AGA over here, arginine. So putting it into a whole different row of the codon table. Similarly, a deletion, GTA. So GUA becomes GAG. That changes that amino acid and then changes the one after that. Changes everybody after the mutation, in fact. So let's explore this in frame shift mutations. Let's see if I'm allowed to. All right, I got a text tool. It'll be a lot easier. All right, so let's say we have a normal sequence, and I'm just going to put these into letters of English, and we can make a sentence. So you say, the big dog was red. Right, so I've got a sentence of 5 words. Each one is 3 letters long. This would be kind of the equivalent of a coding sequence that has some codons. And so if we insert a letter, let's say we add the B, A, I, T, D, O, right. All right, I took all the same letters, I inserted one, and after that point, if I treat these as words of 3 letters each, everything gets a little bit, it's just unreadable. So the effect on a protein is really that everything after the frame shift mutation becomes garbled nonsense. And so like not a nonsense mutation, but just amino acid sequence that is basically random, it doesn't make any sense, certainly wouldn't produce the same codons as were being produced before. So frame shift mutations have typically pretty big effects as well. We could also delete a letter, the big dog was. So I did an example here where I did a change late in the sequence, and this also could happen in a protein sequence, and this results in something that's pretty readable, except at the end. So it becomes garbled nonsense only in this case right at the last 2 words or codons. That's also a possibility. I mentioned the addition or removal of multiples of 3. You could also do that in an example here. The big fat Dog was red. The addition of, maybe I'll make one that doesn't make any sense. Sorry, not coming up with a good word here. All right. So possibly the insertion of something that doesn't make sense, but the rest of the sentence continues to make sense. So that could actually have maybe a less effect on the function of the protein. So picture a protein with amino acids in the space where we have the words right now. So just kind of an illustration, as I said before, the rule is really, it depends. So as far as the effect of a nucleotide change on the amino acid sequence, and then the function of the protein, that all depends on how important everything was in the pieces leading up to that. All right. So I get this question once in a while. Students ask me, well, what happens if you have a stop codon that mutates so that it's no longer a stop codon, right? So illustrate this a little bit. So we've got our, say we've got our RNA molecule ready to be translated, mRNA, and say it's got a start codon. And put it over here. Stop codon. So if translation usually happens over this length and stops there, if you mutated the stop codon and made a longer coding sequence, so the stop codon mutates so that it no longer says stop, then the translation will translate the nucleotides that still exist on toward the 3' end of this RNA molecule. And again, it's going to be kind of garbled nonsense, doesn't make any sense, random set of amino acids being produced by that. But that might not have a big effect. So this would be sort of a little tail of amino acids added on to an existing, say, fully functional amino acid sequence. So it might not have a big effect. And chances are that at some point after the stop codon, even if that's mutated, there might be a backup stop codon there. And that's what I've got here. This is an interesting result from a study in yeast where they found out that there was a non-random number of backup stop codons after the normal stop codon. So this yeast has kind of covered for the possibility and actually put in their extra, so as a result of selection, put in extra stop codons after the existing stop codon to kind of cover the basis for that. So my photograph here is of a car going off a cliff. That's sort of what happens if you just keep going. You forgot to pay attention to the stop. Turns out that in a lot of cases, you probably run into a stop before too long in that kind of DNA sequence. So we talked about getting mutations and the effects of having a mutation. And now we can look at the process of cells recognizing mutations and repairing mutations. I don't cover this in too much detail in this class. It's a more of a biochemical process, and for genetics, I think we're a little more interested in the effects of the mutation, not so much of recovering from the mutation. This is a nice illustration of one of the ways that cells can detect where the mutation exists. So when a mutation comes around on a strand, generates a mismatch. So you get the wrong nucleotide across from another nucleotide, and it makes a kind of a biochemical bulge in there where G and T are not proper, they're not properly complementary nucleotides. And the problem is that in this case, the cell doesn't know necessarily, looking at a mismatch, G versus T, it doesn't know which one is wrong, right? So it's, they're just nucleotide molecules, G and T. So it's kind of equivalent to if you watch one of these movies where somebody has a clone or a robot taking their place. This is a Terminator 2, where the Terminator has made itself look like the protagonist woman here. They both look the same. How do you know which one is the real person and which one is the robot? Classic conundrum. Well, the cell has a solution for that, is that as DNA gets a little bit older, it gets added, it gets these methyl groups attached to it. So there's like a little chemical modification on some of the nucleotides, and the older DNA, the DNA that's been around for longer, has methyl groups associated with it. And so in making a choice of which strand to fix, the cell is going to side with the strand that has the methyl group on it and decide that's the older DNA, and keep the T in this case and remove the G. So this is showing the removal of a larger strand and then a replacement of the strand with eventually the correct nucleotide across from that T. So repair is possible. That's an important part of the process. So during cell division, cells copy their DNA, and then they also double check and make sure that they've copied their DNA correctly. But there's a time past which the cell would not be able to know that there was a mistake. And that's basically past this point. Here we've got nucleotides that are mismatched across from each other, showing this jiggly appearance of them not matching up correctly. But at some point, if they stick around long enough, those two DNA strands each get copied into their own double strand of DNA. And the mistake, this mutated version on the bottom here that has AGGC instead of AAGC, that would stick around and that wouldn't look wrong at all at that point. So the cell wouldn't be able to fix it, and potentially it'd have an effect on the organism, but otherwise it wouldn't be something that the cell could detect as a problem. And moving forward, even past the division of a single cell, a lot of times we're interested to know that mutations have been passed on to descendants, right? So I mentioned those differences in the OCA2 gene in the human population. At some point, those were passed on to populations of people that diversified and became large groups of people in Europe and Southeast Asia that all shared mutations. So it takes a lot really to get a single cell mutation, a mutation on the level of one molecule of DNA. It takes a lot for that to get into the next generation of organism. So it requires, as this diagram shows, requires that that DNA that's mutated is part of the DNA that gets passed on to the offspring. So within our bodies, we have millions of cells, and most of our cells, almost all of those cells, are not going to get passed on to the offspring. So if you have, say, you've got skin cancer or something like that, a person who has cancer in their lifetime does not pass on the cancer to their offspring, because that's in a part of the body that's not in this so-called germ line, the cells that become gametes, the sperm cells and the egg cells. So in rare cases where the mutation ends up in cells that become sperm cells or egg cells, that can get passed on. And then that becomes something that makes up the whole organism of the next generation. So whatever DNA you pass on, combined with the DNA your partner passes on, that becomes the DNA that's the foundation for all the cells in the resulting organism. And then so at the top here, I mentioned the skin cancer possibility. This fish has a mutation in some part of its body away from its reproductive organs. Then that would affect the organism of that generation, but not the next generation. Another way to look at that, so after mutation, the mutant cells get passed on as they pass on their DNA as alleles. So this fish is going to pass on sperm cells or egg cells to the next generation. And that's only half of the genetic makeup of the next population. So mutation in one of the chromosomes let's say we have the mutation called A prime here. So A for normal and A prime for the new mutated allele. This fish would have only one of its two alleles for that gene be mutated. And depending on how that gets passed on to the offspring, it would pass on either the A prime or the A. So this is kind of one of the benefits of being diploid, is that mutations don't always get passed on to offspring. And mutations that say they're harmful or something, they might be selected against in the population. So even if there is a pretty rare chance here, even if there's a mutation that affects the germline, it would still only get passed on to half of the offspring. And then as a diploid, we can ask the question of what happens for a diploid organism for its phenotype resulting from one of these mutations. So this is another one of those that becomes an open question. So not only do we ask how the protein is affected, we can also ask how that protein contributes to the phenotype. And so we can have mutated alleles that are dominant, mutated alleles that are recessive, mutated alleles that have no effect at all. So I just want to finish up with some examples of ways that mutated genes can contribute to an altered phenotype. Sometimes they're referred to as loss of function or gain of function. So a mutation most often produces this kind of effect. So it's much easier to break something than to accidentally make it work in a good way. So an example here, this is a mutation, an allele in the human population that causes color blindness. And if an individual has the mutated version, then they have this rhodopsin protein that doesn't perceive color. So it's mutated to be effectively broken. And if a person is homozygous, or this one is actually a case where it has some sex linkage, a person can have only one copy of the X chromosome and have that be the mutated copy of the rhodopsin gene. They would be colorblind and maybe not be able to see the color that's buried in this dot diagram here. Oh, there it goes. Okay, so loss of function, and that would contribute in this case to a recessive allele. So just be a recessive allele by losing the function, meaning that if a person was heterozygous, then the dominant normal version could cover for this deficiency. One of the things that's, I guess, more often comes up in lab, if you want to really get at like most extreme version of what happens when a gene is mutated in the lab, you can remove the gene entirely. So that's called a null mutation or a null allele, null for like nothing. So this is an example of a gene called BZ here, and in the laboratory they deleted it, and they can do that and then see what the effects are. So the fruit flies that we have in lab that have mutants, those are null mutants for the most part. They're just completely missing whatever gene it is that's under study. So the brown mutants are mutated for the brown gene, where they're missing that gene entirely. And you can see like the full effect of missing that protein by not having any protein at all. And the second bullet point here just mentions that under natural situations, you could get that effect, basically the same effect of a null mutation if you had an early stop codon or one of those early frame shift mutations potentially. It's also possible in a lot rarer cases for mutations to do what we call gain of function. And my example here is also from color perception in humans. And it turns out that we have these three main kinds of color receptors: blue, green, and red. That's what they're called anyway. And the green and red diverged as a result of a mutation. There's a duplication, so they had two copies of green, or whatever the ancestral one was, two copies of that, and then those two copies diverged and became a little bit different in absorbing different wavelengths of light. So actually gave humans 3-color vision as a result of generating a new version of this opsin protein. So this is kind of a schematic of an opsin protein, and there are several kinds of that. So we have these 3 color receptors. They're all like related proteins as a result of copying. Here's a phylogeny if you wanted one. So humans and our ape relatives, as well as old world monkeys, these are primates that have that 3-color vision. So the ancestor of those groups, way back at the ancestor of apes and old world monkeys, they have that gene duplication that gave them the green vision, gives 3-color vision. Thank goodness for that. We can see 3 colors and perceive a lot richer range of colors as a result of duplicating a gene and having the 2 copies diverge into what we have now as red and green colors. So that would be a dominant allele. Having that ability, that's a protein that has a function, so gain of function. And a person, even without the duplication, I suppose ancestrally, you might have an individual who could have the red version and the green version and pick up a new color perception function. A little bit rare. I had to search for this kind of example, a loss of function allele that is dominant. So this becomes a dominant allele, but also has a loss of function. And the effect in this case is a gene called FBN1 that can be mutated so that it actually interferes with the function of the normal FBN1. So it's kind of a negative effect. And a person can have that negative effect by being heterozygous for this mutant because it messes with the normal function. All right, last topic here is evolution. So as DNA changes, some of those DNA changes get lost as a result of selection. So if an organism is missing some ability that affects its survival and reproduction, a little bit more in a natural context, not as much in the human population, but if you can imagine an animal that loses color vision and cannot perceive food or predators, that would affect its survival. That might cause that allele to be lost. So individuals that have mutated genes that are missing functions, they get eliminated from the population, and along with them, their genes get eliminated as well. You can actually see some of this signature is basically looking across the DNA. You can see the relative importance of some pieces of DNA versus others. And if you line up the sequences for gene regions across individuals or across different species, you can see there's some regions that are what we call conserved. So this area here is completely unanimous across all the sequences here. And this is a ribosomal RNA gene. Over here is another ribosomal RNA gene. And they're very similar to each other. And in the space between them, that's just a spacer. And it doesn't have a major function. And it has been allowed to change quite a bit. So there's a lot of difference in this region among individuals because the function of that is less important than the function of these ribosomal RNA genes, which if you mutated those, you wouldn't have a functioning ribosome. Kind of difficult to get by there. So just highlighting here, this is the coloring now shows blue for areas that are the same as the top sequence, and yellow for sequences that are different from the top sequence. So regions with more yellow are more divergent from each other, and regions that are more blue are more similar to each other.